Natalia for Utah State University, and it's my pleasure to introduce our first speaker at today's workshop. Jim Reese is a professor in the Department of Animal Science at Iowa State University. He's had a long and fruitful research and education career in the areas of cow genomics. He now serves in administration at Iowa State University as the Associate Vice President for Research and the Director of the Office of Biotechnology. Jim has been active in NRSP 8 and very successfully fills the role of the NRSP 8 Coordinator for Bioinformatics. A special thanks goes to Jim because he is the PI on a NIFA grant that has led to the high throughput phenotyping and big data analytics workshop that many of you attended on Monday and Tuesday. And he also included in that grant the outline for the Genome2Phenome workshop that we're attending today and tomorrow. Jim's assignment for today's talk is to summarize what happened at the high throughput phenotyping workshop from Monday and Tuesday. So Jim.
Thank you, Noelle. It's not every day that you get to come to a meeting and have a president of a university actually do the introductions. But it also says how much dedication Noelle has for this general area, which is great to see. Thank you, Noelle. So over the last day and a half for starting this, we had 96 people who had signed up for an area and a topic for high throughput phenotyping and data analytics, which is, I think, beyond any of our organizers' wildest dreams that we would actually have that many people here. They represented universities, federal agencies, industry, as well as several international folks for giving different talks and the discussions thereof. And they represented really all of the different species and multiple disciplines, which was great to see. What we wanted to do was really discuss really more state of the art for high throughput phenotyping. What are those things, what are the opportunities that are out there for us to collect phenotype in a different way than what we've done? Jeff had mentioned that to some extent, that this is a place where we probably are lagging. From a genetic standpoint, we do a great job of genotyping. It's become relatively cheap in comparison to where it was. And hopefully we have additional leapfrogs in technology to drive that cost down even further. But what do we have to do in order to commensurately bring that phenotype along with it, such that we can make genetic progress in a lot of different traits of interest? But at the same point, how are we going to handle the data that is associated with this? When we deal with simple genotype, that's relatively simple. It's a lot of data, don't get me wrong. We got 2 million plus dairy cattle that are genotyped. But when you start to add 2 million pictures on top of that, the order of data goes up by a magnitude, if not more. In my own mind, it was somewhat that we were species agnostic, because this is something that is needed for every species. It doesn't matter if you're talking about chickens or aquaculture. Now, how we're going to address the problems in each of the species probably will become somewhat species specific, because what we're going to invest in an individual poultry bird may be a little bit different than what we would do on a $250,000 dairy cow in terms of doing stuff. So there will be differences in what we would do. Hopefully, it would also be discipline agnostic, that we can incorporate from a genetic standpoint all of the nutrition, all of the physiology, all of the meat science into the things in which we're doing as well. So can this help us to some extent expand our ability and impact as a genetics community on animal science? What we tried to do is identify some of those opportunities. So there were the talks as well as then some breakout sessions. And hopefully we tried to say, what were those gaps? And I'll try to explain that a little bit as to what we tried to frame it with at the very end. And then articulate where we would try to go for the future. And our desire was to have a marker paper that will get published. We've started to work on that as a general outline. Our hope is to move it most of the way towards publication by the time PAG rolls around, that we can try to report more of that back at the NRSP8 meeting at that point in time. Also when we talk about the outcomes from this meeting as well. So from an agenda, there were 8 different speakers, both plant and animal folks. They range from technology to application, data analytics, data analysis, data visualization, portions to it. There were 6 additional lightning talks in the breakout sessions as well. And we did this all over the day and a half. To give a little feel for some of the different talks that were out there, I just got one slide from each of the 8 main in order to try and point out some of the different portions that they had. So Natalia della Leone gave the first talk. She was talking about the Genomes2Fields project. This is a huge project. Over 18 states are involved in it at this point, trying to link genotype and environmental interactions together. And one of the things in which she talked about was simply scanning either seeds or the cobs or the tassels in order to collect phenotype on a scale in which you'd never do individually if you had to measure it by hand. And the point which she tried to make is that they collected these images and they keep going back to these images because there's additional information to be gleaned from it that they did not realize when they first scanned the seeds or the cobs or the tassels, or the other portions that you would have for growth of the plant and other things. So there's a lot of data reuse that occurs in this project as well. Santiago Verdugo from Avigen had talked about the need to bring in new phenotypes, and one of his main points that he had is in reality there's no room for error when you try to incorporate it directly into their breeding program. You got to know that this is something in which you absolutely want and you need in order to move, in their case, the flocks forward in order to meet industry needs. One of the things from a new industry need that he pointed out in the US is the fact that we now have, for broiler production, about 40% of all of the birds in the United States do not ever have any antibiotics given to them in order to meet a consumer demand. And this has changed what they have to do in terms of selection of those animals in order to maintain the welfare. Victor Zavala from the University of Wisconsin-Madison was talking more about a systems-based approach. So he came to us from a chemical and biological engineering. And what they're trying to do, at least within Wisconsin, is to map inputs going into the dairy industry and where they're coming out in terms of the manure and where those nutrients are actually going, and how to try to, in essence, develop a system to manage everything in order to improve the environment and other portions that they have. So he thought about it in a slightly different context than most of the folks. Raluca Metesky was talking a little bit about a heat stress project in which they're running at the University of Florida. And in this case, they were looking at sweating rate as well as time in the chute and the effect of just simply the time of day. When you're early in the day, they're going to be cool. There will not be as much sweating the later you go. And what are these things that we have to start to incorporate into our models in order to deal with something that is purely just a management? It's not due to the genetics of the animal itself or some treatment portion that we put on it. We have to think of those things as well as we expand our ability to handle the phenotype. Nora Bello from K-State did a really nice job of trying to let us know that we still cannot forget about experimental design. Just because we have 1,000 animals doesn't mean that we have 1,000 independent data points. Nor does it mean that we have any statistical ability to make inference from the data in which we have. So think about what it is that we're collecting, why we're collecting, and how we would collect it in order to control for things as we go forward. Liang Dong from Iowa State University, it's a little hard to see, but there is this little strip right here that is what they're calling a wearable tape that they can put onto the leaf of corn and measure respiration that is taking place. How much water, how much CO2 is coming off of this leaf, and do so over a longer period of time. And this tape is actually something that's stretchable. So as the corn grows, it just stretches with it. And you can continue to collect phenotype for it. So what are, in this case, some of the different new technologies that we need in order to collect phenotypes in a way that we've never done? Is there something that he also talked about literally having a 100-micron probe that you stuck into the stalk of corn in order to measure the nitrogen uptake or phosphorus uptake into the plant itself. Are there new technologies, new sensors that we need to develop in order to measure things that today we don't even comprehend what it should be or could be for moving forward? And then Dr. Hart talked about how to try to visualize data. So he runs a large MOOC out of the University of Illinois on how to graph in order to give information back. And the example in which he gave here was all of the different football teams playing each other and how you can literally start to find all of the conferences and what conferences tend to have more of an agreement between each other for looking at things. But how you visualize it has a big impact on how you are able to physically interpret the data that is there. So that was the 8 main talks in which we had people gave. They were very varied across, but also one fed upon the other in order to give us a broader view of what were the possibilities for going forward. And in the breakout sessions, and we're trying to summarize this, so Nick Sorrow did a great job at the end of the meeting trying to summarize it. Now I'm trying to do as hopefully a good a job as what he had done. For all of it, at the end of the day, we're there in order to say, what can we do for livestock production? What is it that we can do in order to help industry supply food for the world, improve animal welfare, all of the things in which it is that we want to do for livestock? That's what everything goes back to. How do we need to do it? Well, one of the problems in which we talked about for the high throughput phenotyping and data analytics is we need to have, in essence, to some extent, a paradigm shift in the people. What is the knowledge base in which they are comfortable working in? Whether it is communicating more effectively with the engineers to build a better sensor, or to handle the data in a way in which they were not yet used to. So things in which we can do to build our own human capacity for handling, developing, collecting this information is extremely important. We also talked in a lot of it that went back to technology. So I put up here sensors that were developed for different things. Well, it's not just the sensors themselves. We need to have plenty of that. But we also have to have new technology and how to physically analyze the data. How do we share this information? Are new technologies as well. And then it came down to eventually the data itself. What is the phenotypes? How is the integration? What is the analysis? What is the visualization that was there in order to help people to better utilize it in order to go back in order to improve livestock production itself? And then there was an interplay between all of these. You can't do one without the other. So we have to start to think to some extent about things in potentially a broader scheme than what we have been doing to some extent. And in order to bring that point forward, one of the things we talked about is really trying to facilitate things all the way from an individual experimental level to something that is more systems-based. How can you explain all of, in essence, dairy production and all of the inputs, outputs, and movements thereof? And in order to try and help visualize this to some extent, back in the mid-60s, there was a paper that I read, and I encourage everyone to go back to it, that is called Bricks in the Brickyard. It is talking about why some disciplines make dramatic improvements while other disciplines do not move so fast. But at the same point, it says that we have to allow individual experiments as well as things that integrate to a greater extent. So from the bricks in the brickyard, we have done plenty of experiments. I've done lots of them over my time. It is really almost in essence just turning the crank. I did the same thing that I did last time because it's exactly what I know how to do. It's easy, it's fast. It allows me to get all of those things for my career that I needed. I had to have publications, I had to have papers in order to say that I would get promoted. But at the same point, we can start to put those bricks together in a better way to make something that is now functional. Whether in this case, maybe it's just a wall. We're going to wall the world off in terms of doing it. So some of the projects that we have for CAP projects and others are bringing different bricks together to make something bigger than what they were. But ultimately, what we've got to move towards is some large edifice where all of this information can feed together. And one of the points in which we talked about was for this high throughput phenomics and data analytics is you really have to have the ability to take everything from individual bricks. You've got to facilitate individual experiments. And when you can, the ones that come together to a greater extent. But can we then reuse that data in order to integrate something to a greater extent than what maybe any one individual experiment could physically do in order to move the field forward? Now, to do this, it's not going to be done just simply at the level of an individual animal or even population of animals. We also have information that we need to collect at the level of a tissue. So I'm showing my own personal bias, having done a lot of growth and development, primarily in skeletal muscle, that we have myoblasts and skeletal muscle there. But you can collect phenotype at individual cell level, at a tissue level, at the animal level. All of it is useful for moving forward. You could look at it in several different ways of what you would physically want in terms of continuums as well. Nick tried to summarize all of it as well as saying, okay, we've got data, we've got other players that are there as well. This isn't something that we have to do all ourselves. There is plenty of work that has been done on the human and the plant side that we can draw from for the information in order to move the science forward in the applications that we have for livestock themselves. Now, some of it, as Karen said, we don't have to really worry about ourselves at this point, but at some point we're going to need some funding in order to try and move some of this forward, because these are not simple projects. So with that, I need to have a lot of thank yous for earlier things in which Karen had said, so the Ag Library itself, for the space in which we were in, for USDA-ARS, for Karen, for Janice, Lauren, Kimberly, and then NIFA, as Noel had talked about at the beginning, were kind enough in order to provide the funding for that meeting as well as this one. So Lakshmi was very instrumental in trying to push myself, Guilherme, and others to move forward for this. And then the organizing committee without Guilherme, James, Nick, John Cole here or not? I don't think I've seen him this morning. And then Raluca Metesky as well, who helped organize everything. If it wasn't for their great deeds, this whole thing would not have come forward in terms of having a group on this discussion. With that, I try to answer questions because I figured that we needed to have plenty of time for that.
Not a question. I think you missed Tammy's presentation. So computer vision will be a very important contribution in the phenotyping, right? For
Tammy Brindle.
From
Oh, Tammy! Oh, shoot! Yes, I did. Thank you for saying that. Yeah, so Tammy had talked to us about actual collecting of images on sows and looking at what size they were, what were impacts that they had on the piglets that were there, social interactions. I don't know what happened to that slide because that was a great slide. She had one that was in pens, literally showing how the pigs were moving around, and she only showed 4 out of the 20, I think she said, because when you got to 20, it was just, she said, incomprehensible. But even at 4, it was amazing how much the pigs would move around. On average, they were 4 kilometers a day moving around in a pen. And someone asked, okay, is that just because they were shaking their head? And they were like, no, you actually had to move at least a half a meter before it would get measured. So there are all of these other phenotypes when you start to put some of these sensors on animals that you will get that who knows where it's going to lead. So thank you. I don't know what happened. I know I put that slide in there at one point in time. Nobody. Yeah, Jerry.
Thanks, Jim. So my concern is that we sort of don't get into a mindset that it's sort of phenotyping for phenotyping's sake, right? Just gathering data because we can gather data, or, you know, there's a huge amount of data that are possible to gather, but NIFA is really, you know, a limited funding agency, right? And if you particularly read some of the documentation that we've got in front of us, the things that currently come up are traits that have been historically important, and they're as Ronnie said, the difficult to measure but economically important things. We're talking feed efficiency, we're talking disease resistance, and we're talking ways to measure them that can be done in a high throughput, so perhaps looking at indicator traits. So maybe you could sort of talk a little bit to the prioritization of phenotypes. You know, we've had a lot of data here that have said, look, here's all of the phenotypes that are possible to collect, and you didn't address things like medically relevant phenotypes, you know, metabolic diseases and things that could have inference across humans, reproductive disorders that could also extend across human populations. So I think there's a real need for us as we go through this process to talk about what is the prioritization for investment in phenotyping.
You bring out an absolutely wonderful point, Jerry, in terms of doing it, because if it doesn't go back to the animal and for some practical purpose to start with, why are we doing it? Why are we spending time on a phenotype that does not have something that is tangible at the end? Now, there may be some added benefit because we happen to collect it for one reason. It also applies to something else. So there is an added on. But for each of the different areas in which we are, whether it's disease resistance, what is the right thing to really measure to predict the ability of an animal not to get sick? We've had groups that have worked on that. Holly has been, and yourself, involved in the BRD project. Do we have the right phenotypes for that yet? We got, all right, it's dying again. Oh no. But from a nutrition standpoint, what is it that we really need to measure in order to optimize protein, amino acids, micronutrients? So every one of the different areas throughout the entire growth cycle or production cycle in livestock, there will be different phenotypes, but they have to have a reason why we started to collect them. You bring up the point of using more medically relevant traits. So if it is in the horse for the stuff that Molly is doing for metabolic syndrome, they would have different sets of phenotypes there that you would have to prioritize for looking at. So the space in which you have is huge. I mean, there's no doubt about it. But if within a given area of inquiry you don't have some priority, because you just can't measure everything. I mean, I would love to be able to measure the entire metagenome, transcriptome, metabolome on lots of animals. No one's going to give us enough money to do that. It's not going to happen. We do have to have priorities, absolutely. And this also brings up the issue that I touched on a little bit about data sharing and a transformation to some extent that we have to take place there. How? Well, not how. If you go back a couple years, there is a Holdren memo that says for federally funded data or projects, the data is going to have to be made publicly available for reuse. How many places have something in order to make the data physically available today for an entire project, along with all of the metadata that anybody else could pick it up and physically use the project in which you've been working on? I sure hope nobody raises their hand, because I don't think it exists. But it is something in which we are going to have to move towards in terms of doing things. And if there is the ability to reuse data. So a simple project in which we did, in my own lab, we had a visiting PhD student from Iran. He downloaded all of the microarray data from beef cattle and dairy, I think, or beef and dairy, that was in Geo, and developed networks directly from it in order to look at what is changing from a diseased animal to a healthy, or what were networks that were conserved across all of the traits. That was a project in which we did zero animal work. We simply took all of the information that was in a database. There is a time that I think we are moving towards where we will have to have more and more people in order to help analyze data, in that we are collecting it faster than what any one group can physically analyze it. And we probably all have data sets that I wish we could do this, but I don't have a physical person to do it. Not everyone has to collect the phenotype, not everyone has to do the data analysis, but there needs to be more sharing. But this also means that we have to change how we think in order to be open to that change. Yes, Jeff?
I just wanted to say, I think tomorrow we do have a presentation from the National Ag Library on Ag Data Commons, which is a repository.
Yep. This is something which the Ag Library is trying to help move us in that direction. Kirk?
So along with data sharing, something that we didn't talk about but has been discussed in the past is sample preservation and sample sharing. And I don't know whether Harvey wants to bite that bullet off or talk about their opportunity to contribute some resources in Fort Collins to that. But that's another issue that goes right along with data sharing.
Mm-hmm. Harvey, you want to say anything now? He's going to back off. But you're absolutely correct. And this is something for the FANG project, which we're going into a realm where we're trying to annotate the genome to a greater extent, is every project banking samples back on every tissue, so we know that if we have to come back to it a year, 5 years from now, that it's still physically available. Has everyone put the time and thought into doing that? Anything else?
Jim, just one other thing. When you were talking about nobody would ever give us enough money to measure the microbiome in every animal kind of thing, I don't think anybody would have ever thought that they could measure respiration rate on every leaf in a cornfield. So, we also need to change the way we think about measuring the phenotypes.
Yeah, I mean, if we can have a step change in technology to where now it becomes pennies instead of $100 to do, then yes, absolutely, everything comes. So if there is strategic investment in places to identify the 50 metabolites or the 1,000 bug species or genes that you need to do and be able to do so very cheaply, it would make a huge change. One of the points on which I did forget to talk about, and thank you for saying what you did, Jeff, is that we really will have, for the foreseeable future, probably some populations that are really deeply phenotyped, where you can do it. But it's going to have smaller numbers because of the cost today. And hopefully that will then allow the technology step change to allow to go to something that Jared Deckard talked about as being more of a community sharing or sourced project where you now have lots and lots of animals because you have lots of collaborators, but you may have much fewer phenotypes because you've identified these are the things that are the most point that Jerry had talked about earlier, that these are the things we have to measure because they're the ones that have the direct impact upon the trait, and those are the phenotypes that we then need to feed back into the genetic evaluation programs that are out there in order to move the industry forward. So, what else? Yeah.
Yeah, one thing I took from the workshop is that a lot of this information and this data that could be collected, we should make every possible effort to incorporate it into management decisions as well. Because there might be some traits that we always wanted to measure but we couldn't, or it was very expensive, but there is also traits that we've been measuring for a long time, but now we can measure them in real time. For example, instead of weighting animals, we could use images to infer condition and weight, etc. If that is adopted at the management level, will generate data and decision making in the short term, but in the long term, we will amass enormous data sets for doing selection.
Yeah, JP, I mean, it's a great point. I tried to talk really today just about the application for the genetic component, but this data, if it's collected, it should change management practices. Because if you could implement a heat stress mitigation program at a more effective time, or if you could identify that sick animal days ahead of visualization, it will have an impact on antimicrobial resistance, because we could use a first-tier antibiotic as opposed to a third-tier antibiotic against it. So I think it'll have great applications, again, as long as we collect the right things in order to do it. One of the examples in which you talked about, and that Kurt was wondering if we got the right numbers associated, if you got a trait that was 25% heritable, we could explain maybe 80% of the genetic variants today with SNPs. If you have the phenotype, can you do all of more of the environment than what we could do today? Can we push up the amount of phenotypic variants that we can explain from a management, from a genetics, beyond 80%, 90% plus? And if you could do that, it's going to have a dramatic impact upon all of the industries in which we work on. Because now you could predict to some extent, or hopefully move in what we're talking about here from genotype to phenotype to a greater extent. Because we still have to deal with all that portion, which is the non-genetic. How do we deal with all that variation? How do we predict it and incorporate it into the management of the animals themselves? Absolutely, JP.
As we saw with the plant, and one thing we saw with the plant sensors, they also have soil sensors, for example, so you can measure the microenvironment of each plant. And by modeling the microenvironment, something we cannot do now, you can remove a lot of that variation.
So did I miss? Okay. Kurt, go ahead.
So along with the crowdsourcing model, we've also got models in dairy cattle in particular, but more so in other species as time goes on, of producers paying for genotyping and phenotype collection because they are either interested in it as a revenue generator or as a management tool, and leveraging those data sets through private organizations, breed associations, the Dairy Council on Dairy Cattle Breeding, so on, is going to become more and more important. But who would have thought we'd have 2 million animals genotyped in the dairy industry? Not me.
So what you're going towards, Kurt, is all of these bits under data. These lists will go on and on for the parts that are there. But you're absolutely correct. We want to leverage what industry is investing with what are the pieces that we need to do from a federal portion in order to help the whole thing move forward. Because if they're there, they see value from it, they become a massive partner in all of this. It's not that we're going to do this just at a federal herd or a university herds. No, It's going to be with industry directly as well. So, wonderful point, Kurt.
So just notice the training aspect again, and I really liked your example of your student that mined geo, and now we have students, at Delaware that never set foot in a lab because maybe you don't even want them in a lab, but they're computer science students. They really are comfortable with big data, and perhaps in the context of training, we need to think about programs that do emphasize the non-wet aspect of training.
Point that a number of people had made is that we probably have to some extent, change how we train students. We got to get, well, based upon the system and the funding in which we have, we do a great job of training them in an area that is about that wide. When in reality, industry would much prefer that they have this. So what are the things where we take them out of the, give them to Janet such that they're now in industry, working there and get some idea what it is, and then leave there and go to I can never remember the acronym for the dairy portion, but they get an idea from a dairy standpoint as well. And they realize that things are much greater than, oh, I understand the functional annotation of skeletal muscle in a pig in terms of doing things. So we do have to try to expand out. And it is a challenge if we're trying to fund graduate students off of a federal grant, because it goes back to the point that we are all getting judged on how many publications do we have, are we doing what we need to do in order to get that money back into the lab in terms of doing it. So that was another point which we talked about as well. So thank you, Carl, for remembering that part. Yeah, Harvey.
Jim, maybe this is a bit more in the weeds, but in your data sharing scheme up there, why I noticed that you've got legal aspects. Any discussion on that? I mean, because I think it seems that at times this is perhaps a major limitation to the data sharing schemes that we have today.
We put it up there in that it's one of these things that a lot of people need to become more comfortable with in terms of the material transfer agreements, non-disclosure agreements, how we may have to anonymize data to a greater extent in order to allow that sharing to occur. Because we are talking about data that has intellectual property and economic value in and of itself directly tied. But if we can find ways in order to help facilitate that to where it can go to greater extent, it is one of the things I think people just have to become more comfortable and knowledgeable with. We've shown very clearly, I mean, Kurt and Jerry have been involved in this from day one of how to get industry data together in order to push the entire industry forward. And they've dealt with it for a long time. Have we trained all of, in essence, the next generation to where they're effective at handling these same things? But we did not talk about how to try and solve it. Anything, it's just one of those things in which we acknowledge that we have to deal with as we go forward. Because it's not like we have projects that are just purely 100% federal funding, and then the rules are slightly different in how you do it. As soon as we put that public-private partnership together, we have to work effectively in order to keep everyone's interests together. Is there more that you would add towards that for Jerry, Kurt? I mean, you guys have dealt with this as much as anybody.
Yeah, I guess the only comment I'll make is that the more groups you have to deal with, the more of a nightmare it is. So, you know, if we work primarily with the beef industry, and so we're dealing with a lot of the breed associations. They view themselves as competitors when they probably shouldn't. They should view themselves as an industry, and that whatever they do jointly benefits the entire industry, but that's kind of not how it works. And so, you know, they want firewalls around their data, and in some cases, breed associations don't even want to give you the data because they feel like they can internalize their own R&D now and basically go off on their own. And I think that's a very naive and short-sighted view of what the future is going to be for them. So it's not only the academics that need some training here. I think I'm saying that we need to actually work with some of the industry partners and try to educate them in terms of, you know, the utility of their data, in terms of sharing it with academic partners to benefit their own programs and to benefit the industry as a whole.
Yep. Very, very good point. Kurt?
So I'll add to that. I mean, in the dairy industry, we couldn't have accomplished a tiny fraction alone. What we did is a consortium that involved industry, academics, government, Illumina, and I think it's critical to build a group that represents the industry and the needs of that industry, and they'll come along for the ride pretty quickly.
Is there anything, so anyone that was at the high throughput phenotyping meeting, is there anything else in which we have not touched upon that we should relay to everybody else who didn't happen to be here for the last 2 days?
So I just wanted to kind of go back to what you were talking about, the student who went back and just reprocessed existing data sets. With the example from the corn, they also did that, and you touched on it, but I mean, they went back with new analyses and they found new things to look for. So it's the actual saving the original images, saving the metadata so you can come back to it in a useful way for ways you can't foresee now. Like, that's one of the other real potentials that I think, you know, we need to account for now.
If we can.
And along those lines, I'll add, my group kept coming back to data integration. And so we kept talking about if there are ways to standardize information, either from small experiments that allow them to be put together into the bigger picture, or ways to combine information across technologies or across different information sources, that was going to be extremely valuable and win across many different types of technologies.
Yep, there'll be times where we just have to throw data away because it won't fit in with other things because the information's not there. Correct. Anything else you can think of, Carrie? Or any other questions anyone has? Oh, David.
Jim, just one thought I had, and we had it in the breakout groups the other day, is that, you know, we talk about all the data analytics and what we're going to do with it, but the topic of public engagement has not really come up, and we talked about in utilization of this data and acceptance of them. The use of these public funds is really important for all of us, and so I think it needs to be part of the central theme in developing the next blueprint as well.
That was one of the points in which I think that it will be there for the blueprint, most definitely. And it's one of the things I don't know that we really talked about for the high throughput phenotyping, but it's something that, well, Santiago in a way did by saying that they were changing what they're doing for their selection because of the no antibiotics, because this is a social pressure being placed upon what is the products that they are demanding in order to have. So those portions are there and something which we have to be aware of. Absolutely, David. Good.